Hello and welcome to Biology 240.
Hello, this is Nick
This
Cap,
is
and
microbiology,
this is
chapter
microbiology,
3.
introduction
We're
to
now
microbiology,
looking at the cell.
and we're covering
And
chapter
remember,
number
these lectures
2,
are meant to
basically
supplement
looking
and
at how
highlight
we
what
see
you
the
read in the
invisible
book,
world. I
and
just
then
want to give you a reminder
during
that these
class
lectures are meant to
discussion.
be a supplement
So
and highlight
please come
what
to
you read in the
discussions,
book
and we'll
and the
look
information
at
that we
this.
discuss in
What
class.
I want to look at here today,
And so,
the
please make sure
biology
that you
itself,
come to those
is we're going to
discussions.
review a little bit of
What I
what's
want you
going
to
on
know
in
for
spontaneous
this
generation.
particular
This
item
is
here is
the
we're
idea
going to be
that
looking
will
at
not
some of the
die,
properties of light
modern
that
cell
are important
theory,
to understand how we see
and
things.
then we're going to look at characteristics
How do
of
we
what
see the
prokaryotes
invisible?
are,
That is
and
things
then
that are less
what
than
the
0.1 millimeters
of
across,
eukaryotes are.
which is basically the point of resolution.
Again,
for the
remember
human
that
eye. What instruments in
life,
microbiology do we use?
microorganisms
and how
are
do we
things
make
that at some
the
point in their
clear,
life cycle, that is
things
kind
that
of
are
when
colorless,
they are born,
how do we
to
make
when
them
they die,
visible?
that they are less
This is
than
going to be
0.1
materials for
millimeter
Module
across.
5,
They can
Week
be
5 for the week of
cellular,
September 14,
but these are kind of very
2020.
small. They come in many,
what this
many,
figure
many
here
shapes.
shows you are
Okay.
basically two types of
what we want
microscopes.
to talk a
What we
little
call
bit
a
about
bright field
is
microscope
kind
on
of
the
where
left-hand
life
side and a
comes
dark
from.
field
And you've
microscope
heard all this thing
on
about
the right-hand
spontaneous
side. In a
generation.
bright field
And
microscope,
so
we have light going through, and
what
our
this
images
comes
appear
about
dark.
is you get this
And
idea of
on the bright
spontaneous
field- on
generation
the
is when you
dark
don't
field
know where
microscope,
things
we have
kind of
dark
come from,
field, and our
they can just kind of
materials
come out of
are
thin air,
written
and maybe
up.
it's magic.
Some of the
maybe it's
objectives that
something
we
you
need
think
to know
when
for
you're
light
kid or something
is,
like that.
and
So
again,
what
what are
we
the
tend to see in
characteristics
science
of
is, like,
electromagnetic
can we
radiation that are
have
used
a
in
relatively
microscopy,
simple
and how do
experiment
lenses work?
that would prove
and
that
so
this doesn't
basically,
happen?
when we look at light,
And
we
so
look at
here's
a
an
wave.
example of
There's some particles
Francisco
in there too,
Redi
but we're not going to
that
cover
did
that.
this
The amplitude is
a
the
long
height
time
of the
ago,
wave,
and
whereas the
basically you take a
wavelength is
piece
the
of
distance
meat,
between one
and
peak and
boom,
the
you have
next.
spontaneous generation.
And so
These
low
maggots occur out of
frequencies
the
have
meat,
relatively
but if
low
you seal the
number
meat, there's
of
nothing happens in there, and
wavelengths,
people
and
think,
all of
the
the
high
flux can't get in
frequency
there. Has a
and
higher
then if you do
frequency
that, you
in
put some
again, two other terms that we
cheesecloth on
really
top
need
of this.
to
You can
know
see the
are
magnets and the flies up in
transparent.
there.
So
And
some
your,
of the petri dishes
meat
that we're
won't do
using
it. So
in class
what we tend
are
to see, and what we
transparent,
see a lot
plastic or
is
glass
that
we
we
can see
kind of
through.
look for
Opaque
these
are things that we cannot
experiments
see
or
through.
these
And so I
demonstrations
put up here
that
a
would tell
glass
us
of
kind of
milk
what's
is your
going
classic
on
opaque
in
material
nature.
that you can't
And so
see
we're going to
through.
go through
Another item
this.
that we need to know is basically what we
prize
call
that actually
refraction
put all this
and
is-
reflective.
So
And in
you know, again, a lot of
refraction,
people,
basically
even when you
something
say, oh
is
well,
going
maggots
to
don't
kind
come
of
out of thin air,
change
but
its
like
angle.
bacteria
A
can
light
come
beam
out
is
of
going
thin
to
air.
change its
And
angle
the
according
case
to the
of
material that it's
Louis
traveling
Pasteur
through.
this, and
A
he actually won a
reflected
prize
ray
on this.
is
And he had
actually
this
one
long
reflection
swan neck flask, and he
is
showed that you can boil
right of way
media
because
inside
one
of
means
this
another can
flask,
be either
and then you can
refracted
bend
or reflected.
the
Is
neck
kind
of the
kind
flask,
bouncing
and
off a
if
little bit.
materials
And so
can't
here
get
we're
back
looking
in
at a
there,
refraction,
then they don't.
and if you look at the straight pipe,
then
it
nothing
looks
grows
like
in
it's
there.
bent
And
a
if
little
you
bit
kind of
when
break
it
off
goes
the
into
top of
this
water,
flask. The
that
same
stuff
thing
will
happens
still kind
when
of
you
grow inside of
put a
there.
pencil in some water.
Again,
and this is one of the
remember
reasons why we
your,
use immersion oil when we're looking
timelines
at
that you
oil.
guys all made.
You
And
will
so
be
Robin
looking
Hook was
at,
one of them, and we do a whole bunch of different things.
You will be looking at a number of lenses.
You know,
and the
again,
great thing about
scientific
lenses
theories,
is that they
sometimes do
refract
not catch on
light
and,
towards a focal point.
Vic
And
Rao
that's the
and
point
a
where
lot of these
things
guys are
will
talking
be in
that cells
focus.
only
And mainly
come from
we're going to
cells,
be using
don't
the
have
convex
spontaneous
lens, which is in the
generation.
box.
That
Here.
came up.
We will
Wallen
mainly
in,
be using visible
1920s
light
had this
that is
idea
anywhere
of
from
where
400 nanometers to
organelles
700
in,
nanometers.
eukaryotic cells came from. And so, you know, again, we were looking at how did eukaryotic cells
and you
occur,
should have an
that we
idea
think
of
this is
basically what
proto-eukaryote.
these wavelengths
This
are.
proto-eukaryote
Again, light that
then,
has all of
developed
these
a
wavelengths
nucleus
is
and
going to be
endoplasmic
white
reticulum,
light.
and that we think
now,
it
how do
took
we
in some
identify what
aerobic
we see? A lot of
bacterium
times what we
and
want to do
possibly
is
some
we want to put on tags.
photosynthetic blue-green
And so some of
algae,
these beautiful
and that's what gave us
micrographs that
our
you see
kind of
here
modern
we
eukaryotic
have various
cells.
cell structures. They
And,
can either
I think
absorb
Wallen
different
believed that
dyes,
he was
or
able
we can
to,
use antibodies
cultivate
with
some of
different
these,
dyes
which
attached
is
to them
not
so that
necessarily
we
the case.
can light
But we
this
had this
up.
thing where it's a
And
good
here,
idea, and
we're,
it just kind of really didn't
dyeing the
catch
nucleus
on
kind of this bright blue.
for many, many years.
From a little bit of history
Ignaz
from your
Semmelweis,
timeline,
who I love saying his name,
again, we looked at
basically came up with
Anton van
importance of
Leeuwenhoek
hand
and
washing.
his microscope.
And
which
again,
looked,
when he did this, a
like
lot of
this.
people laughed at him, and
again,
they
Robert
didn't
Cook
want to wash
also
their
had a
hands.
microscope.
And people for a
Zacharias
long time
Janssen
laughed at kind of
in the
washing
15s and early
washing their hands.
1600s.
But you know, it
There's
really
some
showed the case, and
telescopes, but
there's a lot of
if you kind
disease and
take a
death and
telescope and turn
destruction
around the
that could
way,
be prevented
it
by
is
just
a
washing your
microscope.
hands.
This
And
is
kind of reminds
showing
me
you here
of just this
roughly the
past
bright
year
field
and us
microscope
not
that we will
wearing a
use in
mask.
class.
And actually, I
Make
was
sure
one of
that
them,
you
too,
know
of
all
kind
the
of people
all the parts,
that we
from
really
the
don't
eyepiece
need
and what
to
they
wear
do
masks.
down
And
to
you know
the
what? Quite
rheostat
frankly, it is
and
a
the
really good,
illuminator. This is showing you here
really good
one of the
thing
things that you're going to be asked is why do we
review
use
your
oil
timelines.
and oil immersion?
Again,
and that
Joseph
is
Lister,
looking at the
Robert
refractive
Cox,
index.
these
So
are
if
all
we put
kind of
a little bit of oil,
mentors
and you guys
or
in your slide
start
should have shown me that. If you put a little bit of oil in here,
what we
what
call
happens is that
microbiology.
light that's going through the glass has the same refractive index
the parts
as the
of
oil, and so your light, all of your
Just
light
like you're
will
gonna know
travel
what parts
straight
of
through your
eukaryotic
sample
organisms
into your
and you
objective.
actually
If you don't
compare
have that, what's
and
going to happen
contrast
is you're
it.
going to have
So a
refraction
typical
occurring,
prokaryotic
and then you won't
cell
have as much
contains
light,
a
and your
cell
image
membrane,
will be
chromosomal
distorted
DNA,
a little bit.
ribosomes, and
In
cell wall.
the dark field
Some of them
microscope,
also
basically
put
what we
just
do is
fimbriae,
we put
fimbriae,
a
and
light
capsules.
stop inside of
And we
here,
will talk
so that
about
basically
all of
the
these
only
and
light
kind
that
of
you
what they
see
are.
is
Again,
light that's scattered
microorganisms
or
for the most
reflected
part
off of
don't
your
really
particular
have
specimen.
a
And so,
big
again, to
difference
see
between them.
microorganisms that are
So,
really thin,
again,
like
you
this
can look
Treponema
at 3
polonium,
different trees, and they
it's
can
really
all
nice to
kind
have
of different
a
shapes.
dark field
In
microscope
general,
in order
in
to
bacteria,
do that. It's one of the main
it's
things that we
basically
do.
you don't
Phase
have
contrast
cocci, which are
microscopes
round, bacilli, which are
allow
rods,
us,
and
I won't get
vibrio,
into that much
which
about
are
what it
kind
is.
of spiral.
What phase
lines.
contrast
And so,
microscopes allow us
again,
to do
know
is
what
to kind of
some of
look
those
at a
common
medium
shapes
without
are
necessarily
for
using a
bacteria.
stain. And so what you do
We're
is
gonna
you
look
shoot
a
two
little
beams
bit
of light
at
in
the
there,
physical
and they're
properties
off a little
of,
bit. And so the
of
refraction
cells
on
and
this
their
will actually
parts.
cause some interference and can
basically
highlight
what we have
or darken
is,
various
bacteria
things
when they're in
inside
a
of here.
solution. It can be what we call
dichoidism,
isotonic, which is the same.
We're looking
And
at
so things
so
leak out
differential
of the cell
interference
as fast as they kind of
contrast
leak into the cell. In which
allows
case
us
the
to
cell
take a
will
look at living
maintain.
organisms
And
that
so
normally
again, we're
looking
looking
at
at
we do not.
20% side.
Again, a lot of times when we put stains on these organisms, that can affect how the organism acts.
Dissolve into a
Solvent,
why we use
sugar
into water, salt.
Another thing is, how do we know what we're
again,
looking
when
at
we have
underneath
a
the
high
microscope?
solute
And again, if you know what you're looking at, you
what
can kind of
happens
point
to
us,
water
but
leaks out of the
if you don't know what you're looking at, and so what we can use is we can use fluorescent antibody tagging.
Actually,
so we have antibodies to a particular
going
protein,
into
and that would light up. And so
your,
our indirect is where we have
microorganism.
antibodies
And this can
against
have
an
an
antibody
case.
that will light
And we
this
use
up.
this
And
a
in this
lot
guy
during
here,
growth of
we can
microorganisms. So again,
light up
when
various
we go to the
things that
hospital,
we
you
want
need to have an
to look
isotonic
at.
solution.
Confocal
when they put it in
microscopy,
your-
we use
otherwise you will
this
have an
again
effect.
is used to
And
visualize
so what
structures
happens
such
to your
as
cells when this happens?
cyanobacterium,
these
cyanobacterium biofilm,
same in a
and
hypertonic
this
solution.
allows us to kind of
water
look at
particles
some of
move
those
out of the
materials.
cell, and
In a transmission electron microscope, we're shooting electrons through a
here's the
sample
cell
in order to
losing
take a
some
look
of
at
its
it.
liquid.
In a scanning
hypotonic
electron
solution,
microscope, we're
gains some of this liquid.
refracting
And if you can
electrons
see
off the
here, if you have a
surface,
cell
and
wall,
we're looking at the surface. And so again,
cell walls
here's
are
the
really
transmission electron
helpful
microscope.
to a cell
The
because they
transmission
actually
electron
prevent the
microscope
cell
is very
from
important that
actually
we cut
exploding
these layers very
when that
thin,
happens.
roughly about 100 nanometers
cells
thick.
actually have
and in the
DNA,
scanning electron
and this
microscope,
DNA
we're
is in
bouncing
what we call a
materials
nuclear
off
region.
the surface.
We call it a nucleoid. It's not necessarily
what we
nucleus
want to look at
inside
is
there.
one of the big
so
things
that's
that
where
your book
DNA
really
is
talks
going
about
to
is
be.
this concept that
The
we call the
ribosomes are slightly
biofilm,
different.
where we
We
have
have
this
what we call
extracellular
70S
matrix that
ribosomes,
kind of
and that is
allows
a,
all these different
they,
species to grow in, and this is
they
basically
weigh a
just
little
showing
bit
you
different than
some of the
ribosomes
biofilm
in
developments
eukaryotic organisms.
of
And so,
Pseudomonas
this
aeruginosa.
came up in the 70s. Kind of what
Here
are
is
these kind of
looking
differences?
at multiple bacteria
Prokaryotic
growing
cells
inside
have
of
various
a
types of
biofilm.
what
So
we
this
call
biofilm,
inclusions.
this is extracellular
inclusion
material in here,
bodies.
and then
They can be
bacteria kind of flat
hydroxybutyrate
inside of it.
We see a number of
every
these.
once in a while we do want to look at
see a number of these things underneath the microscope.
actually atoms.
We can view
and
them.
so we have our STMs.
Bacteria can also go through what we call a development process.
Scanning
and
tunneling
we talked about that in
microscopes.
chlamydia.
Again, your figure, your book gives figure 2.28. So what I really recommend is that you go through and look at those
figures. A spore
figures,
is a kind
and
of
you have an idea of
a resting
what
structure,
particular,
and
microscopes
spores can
are.
actually live for a very long time and dry.
Second thing we want to do is learn about
and so here are these
staining
kind of
our
vegetative cells with
Why
the
do we
spores
want to
inside
stain
of them.
things? It's basically
and when
to
kind of things
differentiate
go
between
wrong, these
simple,
spores are able to survive.
what you want to do in
Spores are
order
able
to
survive
understand
things like
staining
boiling
is to be
water,
able to differentiate
etc.
between simple and differential
You
stains.
need to know kind of
You
some
want
of the
to
properties
explain
of the
some
plasma
common
membrane.
use
The
stains.
plasma
You want to be
membrane
aware of the
is a
procedure
phospholipid
for the gram
bilayer.
stain. And
It's a
so
lot
again,
like
when we
a
talk about
soap bubble.
taxonomy,
Well, within this
the first thing
plasma
that I
membrane,
want
you have
you
a
to
number
do
of
is to tell me
proteins
whether it's
that are
gram
attached,
positive or gram
and then you
negative,
have a
and whether it
glycolipid
can do that
that's,
or not. In order to look at things on a slide, we need to fix our sample to the slide, and we can do this by either heating it up or placing it in a 10% formalin. Basically, what we're trying to do here is, again, we're looking at things like glass slides. We're going to be rinsing and washing them off with a liquid, and so we don't want our sample to go. So what we want to do is we want to stick our sample
can diffuse
and make
through
it
the plasma
stick
membrane.
to our glass slide.
It's difficult.
An example of that would be doing, especially in dishes. I hate doing dishes at night. I always want to wait till the morning. My wife doesn't like to wait till the morning because actually things dry out and they get
bacteria and other living organisms have these transport channels, and so these protein transport channels are water channels.
Slides,
and what this allows is it's a little hole inside of the plasma membrane. Now, it allows materials to kind of get
again,
through.
when we look at things, we can have what's called a positive stain
this is what we
and
call
a negative stain.
Diffusion.
And so when you're doing positive staining, when you're staining an item, when you're doing negative stain, so you're staining everything else around there, and your item doesn't take stains. Trouble is, we also use this. We have what are called basic positive stains. In active transport, what you're doing is you're moving positively charged against a concentration gradient. You're actually using, and then energy again, we have the opposite hydrolysis of ATP to make ADP.
and inorganic phosphate. And we use this to kind of gain some energy.
In gram staining, the gram staining procedure, and I'm going to be mentioning this a whole bunch of times.
Things are alive, they are pumping things across.
Again, gram staining is a differential staining technique that uses a primary stain and a secondary counter stain to give you the difference between basically one of the large morphological differences that we see in the bacteria, which is that
subunits, which are
cell wall. And so in the gram stain, we first, after we fix our bacteria, we add crystal violet. This are the primary stains that actually stains everything blue. And then we add a mordant iodine. Iodine will actually bind to your crystal violet and actually make it a little bit larger. After staining with iodine, that mordant, it's still blue due to the crystal violet. When we add alcohol, the alcohol is able to wash out the mordant and the crystal violet from your gram-negative bacteria, but it's not able to wash it out in gram-positive. It's much more dirty, much more there. We then do a counter stain, and then that's why in our gram staining we see that things that stain purple are gram-positive. Here's that stain pink or red are gram-negative.
We may have on the outer surface.
And so what we have here, if we look at this here, we have a mixture of bacteria. We have some gram-positive cocci, and we have some gram-negative rods that are in
on
here.
Our gram-negatives,
and so again, sometimes when you have pure culture, you may see something that looks a lot like this. But again, this is the gram-negative E. coli and the gram-positive staphylococcus and things like that as well.
Here's an example.
And so you can actually have a material that's here's an example of but if it's contaminated lipopolysaccharide, and sometimes these acid-fast bacteria like Mycobacterium tuberculosis, which some of you did your talk on, guess what this causes?
A type of glycocalyx.
They have a lot of lipid.
So here we have this negative staining and this kind of little white area seen here.
In our negative stains, we can see things like India
again, one of the reasons why you have to do this kind of staining
actually, this is a Bacillus
not have a charge, and it really won't.
Bacillus cereus. And then we see this kind of clear area around here, and that's due to a capsule
are actually
that's
these
around
channels that bacteria can form to
we can also see specialized structures. This is what we call endospores.
Different ways.
And so the endospore staining allows us to see the endospores inside the bacteria.
Actually,
these endospores have
kind of whip-like structure
green. It's
that
very
allows
hard
them
to
stain
move.
these guys.
And basically,
it's a rotary motor,
a very kind of robust process in order to get the stain inside of there. But again, a lot of times we'll see a hole inside of here when.
And again, we have what are called
monotrichous. Some of you actually were looking at this flagellar staining, parentrichus, Bacillus cereus. We see a lot of the flagella in there, and so we add flagella are very thin. What you tend to see is that when these flagella rotate clockwise, when they rotate clockwise, and so have a good idea of what kind of the bacteria do. When you rotate counterclockwise, transmission all come together, and they rotate, and they tend to cut things the bacteria forward. And these clockwise rotations, what we call tumbling, and the running is the counterclockwise. And what we see is bacteria can actually sense a gradient. And so if they're moving in a sugar solution, they will actually go towards the higher sugar using the microscope is
because they will run longer under those
technology
conditions.
for looking for microbes and samples is pretty straightforward. Even today, many tests still look for microbes using microscopes and staining. We're going to be looking at how DNA technology is kind of changing this process, but you can take a look at it. Again, we will be during our talks today, we will be looking at a little bit membrane-bound organelles. And these can be eukaryotic organism, can be single-celled inside of
what I want to show you here
is just basically how we can use kind of indirect staining, so we can have an antibody against a well-defined nucleus, T-palladium cell, and we have a fluorochrome on this. So this allows for things to light up.
We have a structure called the nuclear lamella, and so there's a membrane that surrounds the nucleus that controls what gets into the nucleus
lecture, and I will see you on Wednesday.
Right next to the nucleus is the endoplasmic reticulum. But in the nucleus itself, you have an area where actually the ribosomes are actually, the rRNA is transcribed, and then the RNA is partially assembled inside of there. So you have these ribosomes that are coming about. I liken the endoplasmic reticulum as kind of like a kind of a manufacturing assembly line. And so you actually have rough endoplasmic reticulum, and then towards the outer portion, you have the smooth endoplasmic reticulum, and then this material will go into the Golgi. And so again, when a cell wants to make something, it will make a messenger RNA. This messenger RNA will get transcribed and then translated into a protein inside the endoplasmic reticulum or going into the membrane. So the rough endoplasmic reticulum is studded with ribosomes, and that's where a lot of the production of protein occurs. And the Golgi apparatus is where you see that those proteins are modified and then they're packaged to kind of get to the end. This is a transmission electron microscope showing you another structure that we see in the cells. These organelles are independent of the Golgi complex. That's not to say that they aren't made by this material, but in the Golgi complex, what you're doing is you're having materials, information that's converted into a messenger RNA inside the nucleus. As this moves out into the cell, it is actually made into a product, and then it needs to go where it needs to go inside of the product. We have these other organelles that are actually called things like peroxisomes, that where we see the cell does different activities. Another important part of the cell is just not a bag of liquid. We actually have what we call cytoskeleton inside of there, and so you have a number of fibers, and then so you have tubulin, we have actin, we have keratin fiber subunits that actually make up the skeleton. The actin allows for movement inside the cells. The intermediate fibers allows for the structure of the cells, and then the microtubules allow us to kind of change the overall shape of the cells and for some movement. A centrosome is controlled with two centrioles positioned at right angles to one another, and this allows for movement of the chromosomes. Another organelle that we tend to not see, again, this is not part of the endomembrane complex, and basically what we're talking about are things like the mitochondria or the chloroplast. The mitochondria, we think, is a microorganism that was captured by it, because again, it has its own DNA, it has its own set of ribosomes, and these ribosomes are 70S ribosomes like we see in other organisms, and the mitochondria are responsible for the production of adenosine triphosphate, and again, so are the chloroplasts. And again, we'll talk about those a little bit later. A phospholipid bilayer, so membranes also occur in eukaryotic cells, and they're very similar to the prokaryotic cells as well. One of the big differences that we have in, again, eukaryotic cells is this kind of movement of materials into and out, and we have this process that we call phagocytosis, where a piece of the membrane pinches off around an organism, and it will form a vacuole on that organism. The macrophages tend to do this a lot, and we'll talk about those a lot. We actually have pinocytosis, and we have receptor-mediated endocytosis, where things are kind of taken into the cell by the cell structures. Outside the cell of a eukaryotic organism, we can have something that's similar to the glycocalyx of the bacteria, and we have this extracellular matrix. This extracellular matrix is the glue that actually holds the cells together. It's this collagen that holds the cells together, and we tend to see that. And so what I want you to be able to do is, here's a cartoon of a prokaryote. You need to be able to fill that out, tell me what the parts are, and tell me what they do. Again, here's a eukaryotic organism. You need to be able to fill those parts out and kind of tell me what to do. This has been Nick Cap, and this is your kind of few moments looking at the complexity of both prokaryotic and eukaryotic organisms. Thank you very much for listening to me. If you have any questions, come to the lecture. We will have a discussion on Wednesday, and you could also move on to the discussion board as well. Thank you very much.